A hush fell over the room. Thanks everyone for coming to the second. I was adjusting the slides, and this is now the second um, James and Kathleen McCloskey Bioscience Endowed lecture. The first was Vinky Ramakrishnan, Nobel laureate. So we were hoping to have one last year. Of course, that did not happen. So this year we're actually having two McCloskey lectures. The first will be given by Dieter Zoll, and I will introduce the McCloskey lecture first and then introduce Professor Soll second. And then on the 14th of October, we will have Joan Stites from Yale University, who will also give Kathleen and James McCloskey Endowed lecture. All right. First of all, I'll talk a little bit about Kay and Jim. And Kay is with us today here, and part of their family is here. Some of the children are here. So again, we thank them for their contributions to the university while they were faculty, and of course their contributions to this lectureship. So first, a little bit about Kay. Kay was Jim's partner in crime while at the university. She also worked in the Eccles Health Science Library, and so was very active in the librarian group at the university, and was always part of the McCloskey team whenever we had scientific guests and we had picnics and celebrations of science at the university. So again, I thank her very much for providing the funds to endow this lectureship. So a little bit about Jim's career and his impact on science. Jim McCloskey's, I will tell you, I'll give you a few examples of some publications from his career. First of all, Jim was a faculty member from 1974 until his retirement. So he was in what was then called pharmaceutical sciences, and then was the department was then named to medicinal chemistry. He also had a dual appointment in the Department of Biochemistry, and so that's part of the explanation for the fact that this is a dual lectureship in the two departments, medicinal chemistry and biochemistry. What Jim was known for was using mass spectrometry to determine the structures of modified nucleosides in RNA, and on the left-hand side of the slide here is a summary. This is a poster that Jim and Pam Crane put together. This is a summary of all the structures of the modified nucleosides in tRNA that were in RNA that were known at that time. There aren't very many additions to this. It's quite comprehensive, and it's safe to say that Jim and Pam and their collaborators account for the structures of most of these that are on this slide. So not only did they put the poster together, but their work actually contributed to the elucidation of the structures of most of these. On the right-hand side is their favorite molecule. Everybody has a favorite molecule, and Dieter and I both have a favorite molecule. It's transfer RNA, as you'll hear. And transfer RNA is heavily chemically modified. We're going to hear a little bit later about the functions of some of those modifications. But Jim's lab really used tRNA as its system for to try to tease out the function and the structure of modifications. And just a few of their kind of more high-profile contributions to science are illustrated here, and the arrows indicate the locations of on the tRNA that they're found. So they're involved in various functions of the tRNA, including modifying recognition by synthetases and recognition and then modulating decoding by the tRNA. And a few of them are unique to archaea, which was something that really became a focal point of Jim's research sort of towards the end of the research, was to look at extremophiles and modifications that are found only in archaea. So here's the phylogenetic tree of life as sort of put together by Carl Woese, and there's one paper on here where Jim collaborated with Carl to discover the structure of a modified nucleotide. I'll come back to that at the very end. And then here's a picture of Jim actually in Yellowstone collecting organisms to try to identify RNA modifications that are found in extremophiles that are archaeal in origin. I highlighted a few of the papers. So Jim was quite prolific, almost 300 publications, I think. Jim was a postdoc at MIT and also in France with Klaus Biemann. So here's one of the first papers. Everyone in the audience who's a scientist should sort of take note of this title. This is a great title, right? Cuts to the chase. Applications of mass spectrometry to structure problems, period, right? These are the kind of papers that you want. A few authors, only two, right? An advisor and a student or a postdoc, and a very succinct title. Kay actually, I didn't know about this, but Kay actually notified me that early in Jim's career, he had a paper with E.O. Wilson, right? We should all endeavor to have papers with people that have the stature of E.O. Wilson. And again, a science paper with E.O. Wilson, 3 authors, biochemical polymorphism in ants, right? Period, right? It's awesome. The other 2 papers are just to sort of emphasize that Jim's impact was quite wide-ranging over the whole world, but he had a big impact on investigators at the University of Utah. So here's a very nice paper from Brenda Bass's lab when she was just sort of starting out with Andy Paulson, where they were looking at inosine modification of adenosines. A very nice biochemistry paper with Brenda and Jim. Here's a paper with Dieter talking about pyrrolysine, and I had a little glance at Dieter's slides. We're going to hear more about pyrrolysine today during his talk, but again, a PNS paper, very seminal paper on pyrrolysine. And then back to the paper with Carl Woese, right? Structure of a modified nucleoside in archaebacteria. So again, Jim's interest in archaea. They were looking at modifications in this archaeal tRNA, 1-methyl-pseudouridine, right? So who knows, the RNA aficionados don't count, but who knows what the current importance of 1-methyl-pseudouridine is? It's relevant. It's relevant to the fact that for most of us here, actually, we could probably take our masks off and be safe, right? So 1-methyl-pseudouridine was originally discovered by Jim's lab with Carl Woese in archaeal tRNA, and it's the modification that is used universally in the mRNA vaccines from both Moderna and Pfizer. So 1-methyl-pseudouridine has these really important properties that now have been utilized to provide unique properties to the mRNAs that are used for the vaccine. All right. So then, this is to remind me that now I'm going to switch gears from the McCloskey introduction to introducing today's speaker, Professor Dieter Soll from Yale. So here's my slide for Dieter, right? He didn't know about this, right? So Dieter Soll is currently the Sterling Professor of Molecular Biophysics and Biochemistry at Yale University. Dieter started off and got a PhD in chemistry in Germany, and then he was a postdoc at the University of Wisconsin with Gobin Khurana. And Khurana, the picture with Dieter and Gobin is shown here. This is a little bit later than when Dieter was a postdoc and when Gobin got the Nobel Prize, but I found this picture on the web. And so Dieter was worked in Khurana's lab until 1968, or Khurana got the Nobel Prize in 1968, and quote, for their interpretation of the genetic code and its function in protein synthesis. And so I think it's safe to say that the work of Dieter when he was a postdoc in Khurana's lab contributed significantly to the work for which Khurana was ultimately awarded the Nobel Prize. After a short time as an assistant professor at Wisconsin, Dieter then joined Yale University Department of Molecular Biophysics and Biochemistry as an associate professor, and he's been at Yale University for his entire career. I would say that part of the explanation for Dieter's astounding scientific success is that he was the department chair for only 2 years, right, for 1982 to 1984. So he was able to focus on science. Professor Soll is a member of the National Academy of Sciences and a fellow of the American Association for the Advancement of Science. He's received many awards for scientific accomplishment and education, and I want to make particular note of the Boucher Award, where Dieter was the inaugural recipient in 2002. And so Dean became, obviously he's an outstanding scientist, but he became very interested in training undergraduates from diverse backgrounds, and he received a couple awards for that. And again, he started these programs in 2002, and or in 1971, and it was recognized in 2002 with this inaugural Bouchette Leadership Award, which has become quite a prestigious award. And the Yale University, again, I think deserves a lot of credit for marking the anniversary of Edward Bouchet. And Edward Bouchet was a member of Yale College class in 1874, and he went on to earn a doctorate in physics from Yale in 1876, and he was the first African American to get a doctorate from American University. So again, with that introduction, I turn the floor over to Professor Soll to tell us about recoding in tRNA.
Can you hear me? Ah, good. It's really a great pleasure to be here. I always wanted to come to where Jim was. I visited him in Houston, and I really worked with him, inspired by him initially. Long time ago, in 1971, we wrote a review on modified nucleosides in Science. And at that time, we wondered what will actually happen to all the biochemical, the enzymes that are predicted to happen to form modified nucleosides. Are we in RNA, DNA, etc., etc.? And through all of this, Jim was really the leading light and pushed everybody, including me. Where are we here? Okay. No, I'm not you there. So, this picture, which I have courtesy of Pamela Crane, I have the same picture, but I couldn't find it because I just moved my office in May, moving into a new building. And the worst that happened to me was to really deal with all the personnel files that I had accumulated since 1967. So in that, I forgot or lost the picture. So Pamela, thank you very much. There was a tRNA meeting in Umeå in Sweden. And this is significant because Umeå is the home of Glenn Björk. And what Jim McCloskey did is really to do the structure of all the nucleosides that are modified. And what Björk did, he started the genetics of all of those. So there was really a very close dependence and collaboration because through the work of Glenn Björk, many things got known. And so this was a rafting expedition at this meeting, and you see the waves were quite astonishing. But this I simply wanted to show. What I decided I will do today is non-conventional. Basically, I looked back on whatever we did in the last 20 years or so, and I wanted to show you what I felt was unusual that we saw regarding the genetic code. So I will very briefly touch on many different topics, and at the end have a 6 slides on a very exciting paper that was published last week. So as you know, the code is manifested in protein synthesis, And this example of proletarian synthetase, for instance, where basically the enzyme takes among the canonical amino acids proline and attaches proline to the business end of the tRNA. The 3' end is proline, while the other business end of the tRNA is the anticodon that interacts with the codon, so the elongation factor will put in the ribosome, and the code comes in here. So the amino acid tRNA synthetases are essential to decode the genetic code. What we know now compared to what we knew in 1965, there are 22 genetically encoded amino acids, I'll come to those, and 61 sense codons and 3 stop codons. But I will tell you immediately what I will have in my last slide. You cannot predict how the genetic code is actually organized and working in organisms. There are organisms available where there are no stop codons. There are organisms available that have multiple codons, etc. So be warned, there will be surprises. What was unknown for a long time, but mainly through the work that we did, we showed that there's an indirect route to amino acid tRNA. And these are the tRNA-dependent amino acid transformations. And in the 80s and 90s, we really showed that GLU-ADT is an enzyme that converts, that is eventually important for glutaminal tRNA synthesis, where basically a GLURS that is non-discriminating puts glutamate on the wrong tRNA. And then the aminotransferase is not going to cleave it, but just amidate the glutamate to glutamine, and the same thing is happening for aspartate asparagine. And this is a pathway that you see in many, many different organisms, and you will even see it in human mitochondria, where you still have the aminotransferase pathway in there. So the question that is going to come, and that's why I put it here in the introduction, is what is happening to the mischarged tRNA? It should come out of here. Yeah. So these reactions, these enzymes are thought to precede the amino acid tRNA synthetases, and the question is really accuracy and what's the fate of the mischarged tRNA, because it is mismade tRNA, how does it not get into protein synthesis? And I will complete this, which a work that was done later. The indirect route also is for cystenyl tRNA and for selenocystenyl tRNA. So there are many organisms in bacteria and in archaea where, as a matter of fact, the tRNA cis is phosphorylated by a kinase to, sorry, in this particular case, phosphoserine is actually charged by the separase, a phosphoserol tRNA synthetase, which is the first amino acid tRNA synthetase that is specific for a non-canonical amino acid. And this then is simply the phosphate gets removed, you work via dehydroalanine and have a PLP-dependent enzyme to then make cysteine out of it with a sulfur donor. So this is going on. So you have phosphoserine intermediate, and the phosphate removal is in a PLP-dependent reaction. And this was really shown by Anselm Sorwald, a phenomenal graduate student. Having seen this, it was very clear to me what should happen with selenocysteine. And this was a highly competitive area. In any case, it was known earlier by the studies of Buck that selenocysteine tRNA is mischarged by cell tRNA synthetase, and then the bacteria, one enzyme, will convert it to selenocysteine, while in archaea and in eukaryotes, the serine tRNA sec is phosphorylated by phosphosyltRNA kinase, and then in analogy to this, a PLP-dependent enzyme, Y-dehydroalanine, will make sec-tRNA. And this is mainly shown by Sodria Paliura and Jing Yuan. So with this then, I just want to introduce very briefly what you should know for Biochemistry 101. Selenocysteine, this is the structure of selenocysteine, and you see it's really related to phosphoserine because instead of the SEH group, you have the phosphate group here, while cysteine has the SH group. And we'll come back to the similarity here. Selenium is an essential dietary trace element. If you take in less than 50 micrograms a day, you die, and if you take in much more, you die again. So there's a small spectrum that is needed to for its activity. In proteins, it's found as a 21st amino acid, selenocysteine, which was shown by Professor Stadtmann in 1976. But in addition to the lower pK value of the selenol moiety compared to cysteine moiety, there is also a different electronic activity that you have. And this is supposedly responsible for the superior catalytic efficiency of selenoenzymes. And I will show you in some slides where we now would like to convert cysteine enzymes to selenocysteine enzymes to gain activity. And the last point of the introduction is that basically, in order to overcome the possibility that the misacylated tRNA is channeled, all these pathways involve structures where the misacylated tRNA, even this one, will, within the complex, go to the final enzyme that will convert the mischarged tRNA to the completely charged tRNA. So for to prevent misacylation, you have these structures. And this shows that only the cognate tRNA is released to EFTU for protein synthesis, and is important for reducing mistranslation. And lastly, in eukaryotes, higher eukaryotes, they do have 20 amino acid tRNA synthetases, including asparagine, glutaminol, and cysteine tRNA synthetase that are directly acylated, while selenocysteine has no cognate amino acid tRNA synthetase. It's actually made by a remnant enzyme from the RNA world. Selenocysteine is formed on the transfer RNA. So I would like to rush you through these and ask questions about the versatility of genetic code, just some examples. Can the expression of genetic code change? RNA modification of the genetic code? Synthetic biology towards orthogonal translation systems, the use of site-specific serine insertion in E. coli, a route to selenoprotein synthesis, and the last is a paper we published 2 weeks ago on encoded deliberate translational ambiguity that is encoded in the genome and used. So this simply was discovered by Lord Pratt in a lab, and this is an organism called Acetoalobium arabaticum, and if you grow it on pyruvate, the protein has 20 canonical amino acids, perfectly happy, and if you grow it on trimethylamine, the protein has 21 amino acids, and this goes back and forth, so you have a dynamic expansion and reduction of the genetic code, and this has to do with the fact that trimethylamine is the first precursor eventually for pyrrolysine. And pyrrole proteins are only made during trimethylammonium metabolism when they grow on it. So we wanted to test what actually happens when you do the following experiment. Pyrrolysine was shown by McCloskey in methanosarcina, and pile T is a tRNA for pyrrolysine, and pile S is the enzyme that we further showed with Jim's help, is the amino acid tRNA synthesis, charges it, and basically all we wanted to do is delete this pile T, and when we deleted this, we showed that if pile T is deleted and you grow it on trimethylamine, it's no longer growing, but it still grows on methanol. So what did the proteome tell us? The proteome told us that what you have downregulated now is methanogenesis from methanol, which is a very efficient route, while what is upregulated now is methanogenesis from methyl sulfide, which is far more inefficient. But the organism persists and lives, even though pyrrolysine is removed, and the other stress responses, etc. So organisms cope perfectly well with more or less or fewer amino acids. Another paper that I was very proud of in a way, and as a matter of fact, one of your students asked me at lunch today, is the following, that I call metabolic recoding. And this goes back to a very old method, the supplementation-based incorporation. So if you have an isostructural non-canonical amino acid, isostructural to the substrate, it binds it and it gets misacylated. And this can be done very easily if you have a methionine oxytroph, and you, or in this case a tryptophan oxytroph, and grow it in the absence of tryptophan, then you can put isostructural compounds in, and then you get a protein variant. And this was the PhD topic of Budiza, who was at that time in Martin Street in Munich with Rolf Huber. And basically what we wanted to do then was we wanted to incorporate and replace tryptophan from a normal E. coli strain with thionylalanine. So this is tryptophan, this is the compound we would like to replace it with. And you see this has a sulfur in here, and has really very different properties of the ring here. But it is also formed by the same TRPRS. And so what was done was a Lenski type experiment that we simply wanted to do the evolution and basically did serial transfer experiments in batch culture with especially knocking out every source that would lead to tryptophan. And after 280 transfers, we had what we wanted. The strain no longer had any tryptophan tRNA, but they had this analog tRNA. So the strain is now confronted with the fact that either the strain puts in the modified amino acid tRNA, or it dies and goes under. Our evolved strain goes only 20% slower than pre-evolved parent, and with this, we recoded all of 20,899 UGG tryptophan codons in a genome by simply restricting the supply of the correct amino acid tRNA and use a different one. So I could say much, much more. And what we wanted to do, but this will be done in the future by others, you can simply go ahead and with this and find out by going to compounds that are less and less and less structurally like tryptophan. How much can you take it further? And how much is a proteome dependent on, as a matter of fact, a particular amino acid or not? There are data that are very old from the 60s and 70s that possibly 55% of the homologies would work with other amino acids. We then tried to come up with a question, and Takahito wanted to answer. How is a genetic code is really expressed in nature? Given that we now have all the databases, all the information in it, what can be done? And we really wanted to do this with selenocysteine tRNA, which is the longest tRNA, that is about 96 nucleotides. And we basically developed a scheme, and we went through about 7 terabytes of data, and we basically looked for what anticodons do we find in selenocysteine tRNAs. And most of them, of course, have UGA as the codon, but there are others, as you can see, that are very different. And in green, these are organisms that still live. And we were lucky to be able to do in vivo labeling with radioactive selenocysteine in those organisms and show that we were correct. In the other case, we simply went ahead with a very old test of the benzopyrrologene assay that assays 4-methylhydrogenase, which has an essential selenocysteine in position 140, the active site, and simply saw that, with one exception, all that we wanted. E. coli expressed this and rescued the tRNA seq. So we believe, and let me go first, that as a matter of fact, selenocysteine is encoded by 8 sense codons and 3 stop codons. In mammals, UGA is important and cannot be changed because our very high-level signal transduction pathway of redox has selenoproteins in it, and unless they get made, you will not live. But in the other organisms where you can see cysteines to selenocysteine, arginine, serine, etc., can be converted to selenocysteine because, and I don't have time to go through all of it, you only, the question is, why do we have selenium? And what turned out in the last few years, it was shown that you have selenium to really prevent proteins from oxidation. If you oxidize the normal protein with cysteine in it, the SH group gets oxidized to sulfide, you can no longer biologically reduce it. You can oxidize selenoproteins up to oxidation state 6, and it's perfectly fine. You simply take DTT and reduce it again. So it is actually a way you can change, the organism can change and respond to a change in habitat. So this is where Jim comes in. In transfer RNA, the position that is really very important is a position. The first position of the anticodon is 34. The position after the anticodon is 37. And this is where we have, and I don't have it here, but it's shown in Westhoff and Grosjean's paper, a host of modified nucleosides. And for this particular, let me take this because I can't show you the other thing. This enzyme that makes isopentenyl adenosine We showed the biosynthesis in 1977. But if you look at this week's Nature, Squire Booker has an article on how the enzyme really works with 4 iron sulfur clusters, and you see everything in great detail. So in a way, if I was young, I would go back to all these enzymes and do the biochemistry that can be done now, and its significance. But what I want to show you here is the N30, what you have in here. And this is a thiouridine. And the thiouridine and the selenouridine you see, and you have a thiouridine synthase that makes this. And it has been shown long ago already in the coded days that if you have the modification here with either selenium or the modification here, that you can encode, you can read the 3' end of the codon with A and A very much better than with G. And data by Westhoff have shown that thiou increases stability of codon-anticodon interaction. And 2-thiou-A is up to 2.5 kilocalories per mole more stable than UA, and 3.4 kilocalories more stable than 2-thiou-G. And so if you have 2-thiou, it will no longer reach the G, it will only read the A. So you can, with this, by the modification of thiou, change the code. It's just one example, and you can ask more questions later on that. Yu-Chen Liu was a super postdoc, and she showed, and we fell into this, that sepsis, the enzyme that I'll show you, a 4-thiou synthetase and 2-thiou synthetase, they are all iron sulfur cluster enzymes that have a 3Fe4S cluster, which is essential for activity, and it's redox active. And we know exactly what nucleotides are essential for it, but the biochemistry of it now will be done, hopefully, by others. The question, of course, is for engineering of orthogonal translation systems that incorporate non-canonical amino acids. This is what everybody would like to do or has done in genetic code expansion studies. And the orthogonal components are tRNA amino acid synthetase pairs. So if you can mutate the tRNA, if you can mutate the enzyme, separase enzyme that puts phosphoserine on tRNA cysteine, and if you can mutate EFTU, because EFTU is a quality control element that will only accept the 20 canonical amino acids and not others, you have to modify EFTU that Olke Uhlenbeck showed also. Then you get, with EFTU or modified EFTU, you can then put for UAG, for instance, the non-canonical amino acid in it. And this has been done by all these people here. Tyrrell started with this. And there are variants of synthetase, elongation factors, and ribosomes. And I will tell you at the end, the component that is least, that is the biggest stumbling block for synthetic biologists is the amino acid tRNA synthetase. The ribosome, you can do many things with it, and the tRNA just as well. And this is just a summary. So if you read the literature, the most used enzyme is the Maripaludis, tRNA, pylRS, also the new Alvis pylRS. I don't have time to go into the details. And the other one is a modified Emianashi tyrase that Schultz did, so they are used frequently, but all these others have been made, and that's just a nice slide that Sergei Melnikov made. So reassigning then UAG from stop to non-canonical amino acid, that has been done many times, is basically you need an orthogonal synthetase pair that's pyrolysyl tRNA synthetase tRNA pyl, and pylar is ideal synthetase as shown here. So pylar-S, as a matter of fact, per se, is a very precise enzyme, but the starting point for synthetic biologists has 4 mutations in it, and this then can really, by now we know, probably about 140 non-canonical alpha amino acids can be acylated by pylar-S. And you can make pylar-S variants that charge 12 canonical amino acids. And this is pyrrolysine. And the cheapest, most expensive non-canonical amino acid is proclysine. And directed evolution is being used on this by many people. And the cost is the biggest problem of the non-canonical amino acid. So we wanted to do something by reprogram translation initiation in vitro. And we would like molecules that can initiate translation on wild-type ribosome. And if you look, these are not amino acids. These are non-amino acids. And I joined an NSF CCI Center Critical Chemistry Innovation Program that Alana Shepard's led. And we really tried to do and see what the ribosome will take. However, we had no synthetase to make the amino acid tRNA from the non-canonical amino acid. So we went to an invention that Hiroaki Suga in Tokyo made, and this is flexizyme, that basically in a chemical reaction you can misacylate tRNA. And then you can do in vitro translation by the pure system to get this, and then we got this incorporated, and so this was lovely. But then the real question was, what the hell do we do in vivo? And Jeff was excellent, and just to say the story was, we figured out that the Yanashi tyrase recognition is limited to this. This is F-met tRNA of E. coli. And if you put this together with very little work, you can really then go and make an initiator tRNA that does the following. I have to give most credit to Tom Rajbandari, who in 1994 showed in a series of splendid papers that you can use UAG as an initiation codon. A stop codon can be used as an initiation codon. So we went ahead and tried to do the same now with non-canonical amino acids in vivo, and basically this is what we have. So it worked with the tRNA that is our initiator tRNA, and it put in a non-canonical amino acid. And the reason we wanted to do so is there are very many, very large, bulky non-canonical amino acids. They do not fit into a protein structure because they are too big. However, should you get them at the 5' end, that is, sorry, at the internal protein, internal end, that is very wobbly, so to speak, they will go in. And since we then can use a genetic selection, we then have a way, once you get a small effect, to use molecular evolution to work on amino acid tRNA synthetase that then would really work with them. And this is really what happens here. So Jeff showed that this is working, and the mass spec showed that this is fine. With this, then, we went to initiating translation with non-UAG codons. And we just looked at a number of substrates and we saw that if you actually use tyrosine, UAU, it should work. So we use tyrosine with it, and it works. There you see the data. And the last thing is, Jeff then did encoding 3 non-canonical amino acids at UAU, UAA, and UAG. And this is different from anyone else in the field did, because they worked with Jason Chin, for instance, with quadruplet codons. But here we do this without quadruplet codons, and it works. So this was our contribution to this. In showing as a way that we do EFTU engineering, EFTU is a quality control element to eliminate all non-standard amino acids from getting into translation. So we actually put 6 mutations into EFTU, which then in E. coli, we could do what you see over there and use UAG and put phosphoserine into E. coli into a method that is really used by many people, and we can make any cellular protein with it. However, we then tried something else, and this is taken from the editor of Science, the journal review summary. Basically, how do we chemically modify proteins after their translation, and how to exploit the free radical chemistry to form carbon-carbon bonds between amino acid residues and a functional group. So we had a paper that came out in Science at that time, and basically it showed if you take, and we did this with ubiquitin or histone H3, that you decide where you would like to put phosphoserine into that. And then you can, of course, take by, you see the methods in a minute, this was done by very, very dilute strontium hydroxide together with zinc powder and copper under aqueous solution. We can make dihydroalanine. And as is known, dihydroalanine, of course, is a tremendous radical acceptor. And if you make some methyl iodide type radicals, you can then form the modified post-translation modification. And what we made is this. And the configuration is exactly the configuration of the one that you see in nature. I was stunned when the Science issue appeared, because together with, with our paper was a paper by the Oxford Department of Chemistry and Drug Development Group with 23 professional authors who did exactly the same. They did it, of course, with more applications, and they used anhydrous medium and used sodium borohydride, while we used the other copper acetate and zinc, etc. But this is a method that you can actually use, and it's being used commercially now for many of these reactions. I would like to tell you 2 more stories. And I have been, really for the last 15 years, very interested in silane cysteine. And silane cysteine, you probably know, but let me just repeat very briefly. You need a seq-specific recoding system. TRNA-seq is the longest tRNA there is. You need a seq-specific elongation factor cell B. EFTU or EF1 alpha does not use cellulose tRNA. And what you have to do, seq is encoded by UGA. UGA is a nonsense codon, a stop codon. So you need a recoding of that UGA to the ribosome to say, this is for cellulose and not for stop. And the release factor doing this is RF2 and not RF1, like all the other things that we know. So it is really a different kind, in some ways, of translation system. That allowed us, and I don't have the slide here, to recode in E. coli 60 out of 64 codons with cellulose. All of this can be done. So the challenge was, this is how amino acid tRNA is made, that the amino acid tRNA made by synthetase, if you put it in the ribosome, while for cellulose the tRNA-seq that is made by a special enzyme that converts serum into cellulose is then go with cell B and this RNA element of about 60 nucleotides, which is in the coding sequence of bacterial cellular proteins. So you cannot really shift it without destroying 20 amino acids of your protein sequence. So what we wanted to see, could we simply with a synthetic tRNA rewire the second insertion pathway to just do like this, and EFTU will do it for UAG on the ribosome and not UGA. It worked, but we needed to do, and I only show it because it's nice structures. Seq, sub-S, the human archaeal enzyme looks entirely different from cell A in bacteria. There is convergent evolution, I won't show you more, but we figured out how the tRNA is bound. And then, this is an aside, we got something else out of this search with all the cellulose-cysteine tRNAs. Takahito discovered what we called allotRNAs, because when Robert Holley got a Nobel Prize and put the globally structure up, the canonical tRNA have a T-stem of 5 base pair, except a stem of 7 and a sum of 12. There's nothing about cellulose-cysteine tRNAs have a sum of 13, but you have 5 bases and here 4, so have 8 or 9 base pairs together. However, the allotRNAs do not follow this, and they are different, and they do not by tRNA-seq, the tRNA recognizing program, cannot be recognized. So the 3 allotRNA species that actually happen in a organism called Sylvibacterium bohemicum, notably it comes from Czechoslovakia. So they were leucine tRNAs, but since cell tRNA synthetase does not recognize the anticodon, we then made this as our main element. But before we wanted to do this, we did a collaboration with the McCloskey lab with Arjun and Jingji and Anna, and we got the tRNA structure. So first, to show basically the allotRNA is really very, very much slower in decoding. In a decoding step, this is simply by single molecule fluorescence experiments, and other things were done, but basically allotRNA is different, and this then led to a crystal structure of this, and basically it shows how one of the arms is slightly squashed. And the tRNAs were simply determined by cryo-EM with the ribosome as a scaffold at 2.5 angstroms. This then led to our third generation tRNA UTU system, where this is our allotRNA that has, of course, parts of cell tRNA synthesis for recognition, part of cell A recognition for cysteine formation, and this is formate dehydrogenase, the only cellular protein E. coli that has an active site cysteine and an iron sulfur cluster here. And we could show by the very sensitive Bentham-Wallerschen test that this particular tRNA can put all the 5 cysteines in there. And then two current postdocs developed a development sex-specific reporter, and this is an intine. And the intine, of course, has cysteine in it, and should the intine have cysteine in it, it will simply splice itself out, so that cysteine has nothing to do with what we want to do in terms of protein, but it will put cysteine instead of programmed by UAG. And we can simply look here at the kanamycin resistance. And Christina and Natalie then figured out how the various tRNA molecules we had, how well suited they are for the protein synthesis with cellular cysteine. And we finally found something that is really discriminated against SEC, because SEC is the only amino acid that would compete, because it would be incompletely made cellular cysteine tRNA. So this works. And with this then, we simply went ahead and showed that we can really, in formate dehydrogenase, replace all the sulfurs and with cellular cysteine, and likewise in human ferrochelatase, we could do the same. And the sister sec replacement does not significantly alter the activity, but this will all need to be checked biochemically. And we then had an experiment together with Fraser Armstrong in Oxford, who is well known for his hydrogenase work, and we simply wanted to make cellular cysteine substituted hydrogenases. I didn't tell you before, formate dehydrogenase of E. coli also has a homologue that has cysteine instead of cellular cysteine in the active site. But that is 1,000-fold less active than the cellular cysteine, so you can actually get quite large effects with cellular cysteine. And we tried a protease, TEF protease, where unfortunately the success was also our enemy because the protease degraded itself. So basically, with this, and this was published in PNAS recently, we made a substitution, and one of the variants is an exceedingly oxygen-tolerant hydrogenase, and it really means something to the cellular cysteine people. I have 6 more slides. This is a paper that was published 2 weeks ago, and I needed it published so one of my postdocs can get his independent NSF fellowship. And it's really about pathogenic streptomyces species encoded translation machinery for deliberate translational ambiguity. It's a totally new mechanism of making mistranslation. So an alanine codon GCU inserts proline. this is the second example since the genetic code was solved of one codon for 2 amino acids. The other example was the Candida utilis, was done about in 1989. It was found serine leucine case. And from further work that is not yet published, it's also asparagine that is doing it, and also threonine. So what is happening that alanine tRNA synthetase decodes GCU normally as alanine. But in Streptomyces, alanine is also encoded, like I said before, but in addition, proletarian synthetase has a tRNA that has, we call it Pro-A, has an alanine anticodon that puts proline in addition to the alanine. And whether there is anything that goes with it, we do not know. But I will tell you that of all the Streptomyces, this is only happening in Streptomyces strains, and only in Streptomyces strains that are annotated to be pathogens. And they are pathogens for potato, for instance, and this is a normal scab lesions by Streptomyces scabies. So the potato tuber is becoming not a smooth skin anymore, has these holes in it, and it affects negatively about 5% of the US potato crop. And someone said, well, it's a lot of potatoes. And the grant application that Oscar sent goes to the National Institute for Food and Agriculture, which is interested in this. But whether there is direct relation, this requires a lot of other work. So if you quickly look, proletarian synthetases, there is an archaeal eukaryotic-like in many organisms and bacterial-like. And they are really very different. And you can see this here and here. But there's an enzyme that we call ProSX, which has a different anticodon binding domain. And that is only found in these pathogenic Streptomyces strains. So we wanted to see what does it look like? And basically, this is a proline tRNA of the organism. This is alanine tRNA of the organism. And the alanine identity elements are this and this. The proline identity elements are this and that. So the Pro-A has basically an alanine anticodon and the proline identity element here. And the various identity elements together form a barrier over which translation or mistranslation happens. So it happens that this is a proline tRNA, charged with proline, even though it has the anticodon for alanine. The other, I don't have it here. The other interesting thing was that this tRNA cannot be detected by any of the tRNA methods by bioinformatic techniques, because this tRNA violates the tRNA structure again in 4 important elements. One is this A in this position. This is the only tRNA that has an A in this position, and it's known from other work that U33 is the key for the conserved U-term of the anticodon loop, and substitution of U33 drastically decreases coding efficiency. And we are interested, of course, to see how the anticodon, which is bound by the anticodon binding domain, really can accommodate this. That we'll know. The gene arrangement is that this tRNA pro gene is next to ProRS, or this is for a threonine tRNA. This is a threonine anticodon, and this is an asparagine anticodon, and this is next to a part of another paralog of ProRSX. Unfortunately, the Proletarian synthesis ProRSX is not soluble in E. coli. So the only thing that we had that links this to streptomyces is a very sensitive test with beta-lactamase, where the alanine in position 66 of beta-lactamase is defective and has very low resistance to carbonicillin, where proline in position 66 has high carbonicillin resistance, and there is about a threefold IC50 in there, and this shows it Pro-A, and this with 65P, the number one, and 65A, and you can see that proline is inserted here. So this is the only thing that at the moment holds this to streptomyces, because we did an experiment that I didn't know whether it would work. We compared the sequences and found out the residues that contact G36, G35, and G36, and changed them from streptomyces to E. coli, enzyme that we call 5 mutations, the anticodon binding domain, and that enzyme is doing what we expect. So it is a result that we got with this. The anticodon binding domain enzyme also showed the result here. If you have in this construct linked GFP with S65, A65, and 65P, you can see that the 65P GFP does not fluoresce, but the 65A fluoresces, while you have no problem, with the M-cherry. And then we saw that this is actually happening by looking using the wild type E. coli enzyme. You really find no difference between the pro-A tRNA, no pro-A tRNA and pro-A tRNA, while with pro-A tRNA you reduce the intensity, as you can see, of the green compared to here. And this is working. The mutants worked out in mouse spec. So we find it's a very exciting system, and I could go on, but I would simply like to show you that everything comes together. Here I have a few domain-specific translation components, mainly in the iminotransferase reaction, that actually have more recently taken some interest in antibiotic development. If a new translational scheme is evolved, nature will use it anywhere it might be useful, and not where we or other scientists believe it should happen. Anywhere you find it. And really the genetic code is far more flexible than at least I had thought. And with this, I really have to thank my group who kept me busy. John Matthew Bile, a post-baccalaureate associate, Christina Chung and Natalie Cron, 2 postdocs from Canada, Kexen, a second-year undergraduate at Yale, and Danny Jiang, who is a fourth-year graduate student at the Academia Sinica in Taipei, and Oscar Vargas, who is the author of this paper. And this is Jeff Thorpe, who is working on the N-terminal addition to the code. Amit Badran helped us with an assay. Sergei Melnikov, he was mainly responsible for the paper. He was postdoc in lab, is now in Newcastle on Tyne. Alan Shepherds is the CGEM director, the NSF and CCI Institute in Berkeley. And Eric Westhoff was so amazed that we see by these tRNAs that he will be our collaborator for the next two papers. And I really appreciate your indulgence. Thank you.
Questions for Professor Zoll? Brenda? Yes, yeah.
Brendan. Mm-hmm. Mm-hmm. Yeah.
Is there an amino acid that you can't mess with? Is there something that's much more rigid in its requirements? Is there a lot to think through?
No. If you look, so pyrolysyl tRNA synthetase evolved from an alpha subunit of phenylalanine tRNA synthetase, and as such is very different from tyrosine tRNA synthetase, especially because the active site of tyrosine tRNA synthetase has two phenylalanines in it that really mean that the tyrosine tRNA synthetase that everybody uses is mainly for tyrosine or tryptophan analogs, while you can do many more things with pylorus. Something that I didn't realize till recently is that pyrolysyl tRNA synthetase, this is a story. We had the N-acetyl lysyl tRNA synthetase long ago before anyone else. But Tom Stites did not want to publish it. Tom said, I'm not publishing a crystal structure of a synthetase without the tRNA. And we couldn't do anything, it didn't bind. Until we found that we need that the KD or KM was 500-fold higher or lower. So simply you couldn't do it. But we had a pure sample of pyrrolysine. There's only one that was made by Laura Kissling 10 years ago. And with that, we actually took the wild-type pyloreses, and all of them had the same kind of Km, Kd, etc., Kcat as the other synthetases. So really, pylorus is a beautiful enzyme. We made it degenerate. And then for the synthetic biology, sorry, I'm not blaming anyone. Really, people are interested in showing something. If I can put this amino acid into the genetic code, if it's only 3% active, I can send it to science. But if I wanted to send it somewhere, do something with it, that's a totally different story. So many of the great achievements that you know of or have heard are really great achievements. But to really put them into the real technical aspect, you will need many, many iterations. And I was asked 5 years ago, what would it take to develop amino acid Trna synthetases? We had a paper rejected by Steve Benner, who said, look, why do you believe in computational methods? Ulmer developed this in 1983, and look how far we got, nowhere. So you can't really make an absolute synthesis the way it's there. So I said this can be done, but I would have to have one graduate student for 4 years for each synthesis. Okay, even this can be done. But if I make the one that I evolve in E. coli and put it in B. subtilis, it's not going to work anymore. So there are so many factors that you really have to, for your own purposes, do the many, many, many iterations. And sorry, this is a true anecdote, and this is how Govind Khorana introduced his postdocs to work. And if you want to go read the prefatory chapter to Annual Reviews Biochemistry in 1964. It was written by Otto Warburg. And Warburg, as you probably know, was one of the 7 lab members of Emil Fischer, who got a Nobel Prize. And Emil Fischer also got a Nobel Prize. In any case, Warburg wrote that Emil Fischer saw that he was very good. But he said, critical, important studies have to be done with innumerable experiments, without much critical hesitation. And today it's called high throughput screening.
Okay.
So,
I think we'll have one more question. So if you have a question and you think that Professor Soll will have a succinct answer, then you get to ask him. You have such a question.
sorry, someone,
I will listen.
I don't have my. Tell him, because of all the masks in my glasses,
Question.
I took out my hearing
Let
aids.
me rephrase it, and let's see if I get it right. So the question is about selenocysteine that has a lower pKa than cysteine.
Yeah.
And so what Hanfei is asking is, does the lower pKa of selenocysteine, does that explain the differences in catalytic efficiency? They're both components. It's the pKa, and it's also, I don't know what it's called, the electronegative potential or whatever. Electronegativity is very, very different. So there is a review that Elias Arner wrote probably 10 years ago. And it says, what does selenocysteine do to improve enzymology? And you can see there are some enzymes, like the formaldehydrogenase, which with selenocysteine goes up. We use GRX and other enzymes and showed it's at least a factor of 10. But in that book, in that article, you also see that some other enzymes,
for reasons totally unknown, do not really have better activity. And the question you didn't ask is, why does an organism have a gene for the sulfur homologue and for the cellular protein? You could easily up the sulfur homologue by a factor of 100, upregulate, and don't worry about selenium at all. So there is something else that you will discover.
All right, I think we will leave it at that. There's a reception outside. So please go out and pick up food and a drink and then go outside. Hopefully still warm up. If you have another question for Professor Soll, you can ask him that question at that time at the reception. So let's thank him again for.
This is yours.